Notes I haven't done from tuition. You just because I had exam, you like went
Hmm.
through the chapter.
Okay.
Can you
All right.
please
Okay.
share all
Hmm.
the notes that we have done
Ha.
till?
I will provide you with the notes.
Okay.
Okay. All right, everyone. Switch on your videos. I will do that chapter as well. Don't worry about it. I'll complete that chapter in the class as well, and everyone will have the notes. Okay. Before 1st
Okay.
October, I am planning to complete the syllabus because no one else has their exam schedule up until now. So yours is there. So I'll complete it. Okay. All right, students. So a little change of plans. We will start with biomolecules today. Okay. Instead of neural control because we have a deadline. Okay. So,
The
uh,
human physiology notes, can you send me all of those? Of all the chapters, human physiology, except the last two, till locomotion.
what? Yeah, again,
Can you
please.
s- can you send me the human physiology notes till locomotion of all the chapters? Because my exam is on 7th. Bio exam is.
Okay. All right, students. So today we're going to start with biomolecules. So when we talk about biomolecules, if you break the word, we have bio, we have molecules, right? Bio refers to living, right? Biology, the science of living. So the various molecules or components which are found in the living organism's body, we call them as the biomolecules, right? Mostly, what are the constituents? There is carbon, hydrogen, oxygen, the basic ones. Other than these, there are many more also, but these are the basic, you can say, elements that the biomolecules are made up of. Now, the biomolecules, depending upon their solubility, depending upon their size, right? They are of two types. There is bio-micromolecules and then there is bio-macromolecules. Understood? So, bio-micro, these are the smaller ones. Okay, they are the ones that are soluble in the acid. So, we call them as filtrate. On the other hand, we have bio-macromolecules. These are the ones that are acid insoluble part and we call them as the retentate. Okay. So what are all the various things? See, you see amino acids, simple sugars, nucleotides, then there's polysaccharides. So all of these are biomolecules. These are the molecules that are found in us living organisms and we have to study about each one of their constitutes, right? So this chapter particularly contains a little bit of chemistry, a very tiny bit, basics of chemistry in it. Okay. So amongst the biomicromolecule, we have amino acids, we have simple sugars, we have nucleotides. On the other hand, amongst the biomacromolecule, we have polysaccharides, proteins, nucleic acid, and lipids. Now, furthermore, when we talk about the biomicromolecule, achha, one more thing. See how I've made several stars around the lipid? Keep it in mind that there is something unique about them. Understood? We will discuss. So when we talk about biomicromolecules, so the reason why they are called as micro means small, macro means large, right? So they are called as micromolecules because they have a small molecular mass. They are less by their molecular mass and it ranges across 18 to 800 Daltons. Examples, amino acids, simple sugars, and nucleotides. On the other hand, biomacromolecules, they are the ones that have molecular weight higher than 800 Daltons. But there is one exception, lipids, the ones that I drew stars around. So lipids, they have a molecular weight of around 800 Daltons exactly, the borderline. Understood? So in terms of its size, it's not strictly a biomacromolecule, but the reason why it is kept under the biomacromolecule because they are acid insoluble, just like the other biomacromolecule. Understood, students? Is it clear to everyone? So that is. Now, talking about metabolites. So, metabolites, these are those molecules that are produced by the plants. Let's just say, for example, there are two types of metabolites, primary and secondary metabolites. When you talk about primary metabolites. These are the, you can say, the most basic, the most important substances that a plant cannot, you can say, phantom living without. Right? These are the molecules that are required for the basic functions such as photosynthesis, respiration, protein and lipid metabolites. Okay, this is like the basic, the most important component. On the other hand, when we talk about the secondary metabolites, so many plants, fungi, and microorganisms, they synthesize some other types of compounds other than the ones that they need for survival. These are those compounds that are mostly synthesized for defense mechanism. Understood? Or for storage purpose or for any other purpose other than the main function of the body. So we call them as secondary metabolites. Okay. They are not needed for growth. They are not needed for, you can say, the basic processes of the body. So we see how the examples of the secondary metabolite. So there's pigments. All sorts of the pigments that you see, they're not initially required for the basic, you can say, metabolism of the body, right? But they are important for other purposes, right? There's photosynthesis, there's protection of the photosynthetic pigments and everything. So under the pigment category, we have carotenoids, anthocyanins. Alkaloids are the substances such as morphine, codeine. These are those substances that are used as medicines for us. Terpenes, monoterpenes, diterpenes are there. Essential oils. You people might have heard about essential oil. Lavender essential oil, lemongrass essential oil, right? These are the stuff that the plant produces. Plant doesn't require it, but we humans can utilize it for our benefit. Then we have toxins such as abrin, ricin. These are the toxins that are there. After that, we have the lectins, example for which is concanavalin A, and drugs such as vinblastine, curcumin, like how different types of turmeric, it has anti-cancerous property in it, right? Same way, different things have different properties in them. So that is because of the secondary metabolite. Then all the polymeric substances like the rubber that comes from the trees, right? The gums, the latex, from where the paper is synthesized and stuff. So that is also a secondary metabolite. The plants don't need it as such, but as humans, we can utilize it for our own benefit. So in this chapter, this particular category and their examples of secondary metabolites, they are very important. Understood? It has been tabulated in NCERT, I believe, and it is very, very important. Any doubts up until now, students?
Ma'am, what are lactins? What are lactins?
Lectins. Lectins are again secondary metabolites that are found in the plants. Okay, they are produced as, you can say, a process wherever a plant needs to defend itself to any kind of stress it undergoes. So in that, understood?
Okay, thank you.
So now we're moving on towards the biomolecules, the different kinds. The first one you people are going to discuss is carbohydrates. So these are those molecules that have carbon, hydrogen, and oxygen present. The other names of carbohydrates are saccharides, sugars, right? We call them with all these names. Now, carbohydrates, on the other hand, they are a variety of types depending upon what they are made up of. So students, do you people know what is a polymer and monomer? In general, polymer and monomer? No. No one knows? See, imagine it like this. Polymer is a very big molecule, which is made up of several monomers. So when monomers, these are like the building blocks. Okay, the building blocks, they come together and make the whole entire structure. So the whole entire structure becomes the polymer. And the thing that it is made up of, poly means many. Mono means one. Understood? So the things or the constituents of a polymer is called as a monomer. Is it clear? The reason why I'm telling you this thing is because now when you are going to differentiate the carbohydrates on different, like on the, you can say into the different types. So there has to be some sort of basis of classification. So here you're going to see different types of carbohydrates depending upon the monomeric unit. Like what is the monomer or how many monomers are. Clear? So we're going to start with monosaccharides. These are the most simple sugars. Acha, one more thing. For example, these are monomers. Okay? They have sort of collected together and they have made a polymer. This is the formation of polymer. Now, if you take the polymer and you break it down, then you'll get the monomeric units again. Right or wrong? Right. Everyone agrees?
Yeah.
But for example, if I take this one and try to break it, will it come into several monomeric units? No. Why? Because it itself is a monomeric, right? It is the most basic structure. It is the most, you can say, lower grade structure, which cannot be broken down into any further molecules. So that's the case. So when we talk about monosaccharides, these are the most simple sugars, okay? They cannot be hydrolyzed. Hydrolyzed means break down in the presence of water into any smaller compound. This is the smallest compound. Mostly, it is made up of carbons ranging from 3 carbon molecule up until 7 carbon molecule. Yeah. So let us see the carbon, like for example, if there is a monosaccharide with 3 carbons in it. So the name becomes triose. Tri means 3. Example is glyceryl aldehyde. When there are 4 carbons present, the name becomes tetrose. Right? Tetra means 4. The examples are erythrose and triose. Then, when there are 5 carbons present, so we call it as pentose. The examples of pentose would be ribose, there would be deoxyribose, and these are very, very important because these are the sugars or these are the monosaccharides that are found in our nucleic acids such as DNA and RNA. Then, when the carbon number is 6, so that molecule is a hexose sugar. Example of an hexose sugar is glucose, fructose, right, which has 6 carbons in it. Then the last one, this is up till where the monosaccharides can go. So when we talk about the 7 carbon sugar, that is heptose, and the example is the ketose sugar. Is it clear, students?
Yes, ma'am.
Now, different sugars, they have different common names also. For example, glucose that we know, C6H12O6, it is also known as dextrose. Sometimes it is also known as blood sugar, grape sugar, and cane sugar. So in questions, they can actually confuse you with other names also. You must know that they are talking about glucose only and not any other. So monosaccharide is clear to everyone.
Yes,
Yes.
ma'am.
Very good. So monosaccharides are those biomolecules which cannot be broken down into any further monomeric unit. It is the tiniest structure, ranges from carbon number 3 to carbon number 7. It can have 3 carbons up until 7 carbons. Then we come on to derived monosaccharide. Now when the normal monosaccharides, when they get modified to form different structures. So now the structure that they form is a derived molecule. So we say that is a derived monosaccharide such as deoxy sugar. Now see, this structure that you see over here, it is the chemical structure of the ribose sugar. Okay, how many carbons are there in ribose sugar? There are 5 carbons, right? So starting from here, carbon number 1, carbon number 2, carbon number 3, 4, and here is the 5th carbon. Okay, now on the carbon number 1, you have OH group on the top and hydrogen group at the bottom. Here, OH at the top and H at the top and hydroxyl group at the bottom. Same goes. Now, when this when this ribose sugar, it undergoes deoxygenation. Deoxygenation means removal of oxygen, right? So removal of oxygen where? So at the second position. Okay, second position of the sugar. So then the structure becomes like this. Now, instead of this O being present here, there's only H present. Understood? So now the structure that you have is a deoxyribose sugar. Since it's like the parent structure from where it comes is the ribose sugar, right? So we call it as a derived monosaccharide. Is it clear to everyone?
Yes, ma'am.
So when the ribose sugar, it undergoes deoxygenation, that is removal of oxygen at the second carbon of ribose, then we have deoxyribose. So we say that deoxyribose are derived monosaccharide. Then same way, from different substances, the sugar can be derived, such as amino sugar. So glucosamine. Sugar acid, that is derived from the acidic part. So we have ascorbic acid, we have glucuronic acid, and then there's sugar alcohol such as mannitol. So these are the various examples of the derived monosaccharides. Is there any doubt, students? Anything you people want to ask? Also in this chapter, you may have several doubts. So if you do so, feel free to ask, okay? Because this chapter is like that. It is, I'll not say.
There's so many terms to learn in this chapter.
Terms are there, but once you get the hang of it, it will become easy. It's not difficult. You know, it was like even more so than botany. This chapter was considered as the most dreaded chapter in class 11. The most dreaded chapter. No one liked this chapter ever. You pick up any student from class 11 and you ask them, do you like biomolecules? You'll have the same answer. Everyone will give you the same answer. Unless they are very brilliant, they'll give you the same answer. Understood? And even I was the same. But ever since I've taught this chapter is not difficult. Trust me, it's very, very nice and it's very straightforward chapter. Only you just need to know few terms from chemistry. That's all. Carbon, hydrogen, oxygen, you people already know. The functional groups you should know, like hydroxyl, carboxylic acid, ketone group. These groups you know, na? You might have studied in those some basic principles chapter.
We learned ketones in our last year itself, chemistry.
So up until derived monosaccharide is clear to you, right? You people are not getting scared away. No. Not developing any internal hatred up until now? Happy to. Now talking about oligosaccharide. Now we're slowly leveling up. Okay, we started with the basic type of sugar. Now we're leveling up. So now talking about oligosaccharide. So remember I told you that mostly the monomeric unit, how polymer was broken down into several monomeric units. When we talk about oligosaccharide, they are usually made by condensation reaction, meaning monosaccharide units will condense together and form the oligosaccharide. So these are small carbohydrate, and since two sugars are joining, so the bond formed in between the two sugars is glycosidic linkage or glycosidic bond. The name of the bond is glycosidic. Usually, for example, there's sugar A and there's sugar B. So when two sugars are being joined, so the bond formation will always be from the carbon number 1 of sugar first and carbon number 4 of the second. They will form a bond, and that bond is called as glycosidic bond, and mostly we call it as C14 glycosidic linkage. 1,4 refers to carbon number 1 to carbon number 4, and the name of the bond is glycosidic linkage. Is it clear, everyone?
Yes, ma'am.
Ma'am, can you explain again?
See, for example, I'm saying that oligosaccharides, they are formed as a result of condensation of monosaccharides. So, for example, there is this one monosaccharide, another monosaccharide, when it condensed together, mixed together, sort of formed a bond together. Then the structure we had is an oligosaccharide. Now, for example, if there's sugar A and sugar B, and you people know that sugars are made up of several different carbons. You have 3 carbon up until 7 carbon sugars are there. So when there is a bond that is to be formed between two sugars, it will always be from carbon number 1 of one sugar and carbon number 4 of the second sugar. and this bond will be called as glycosidic linkage. Since it is being formed between the carbon 1 and the carbon 4, so we call it as C14 glycosidic linkage. Is it clear, Aisha?
Yes, ma'am. Thank you.
So that is. Now furthermore, talking about depending upon how many monosaccharides condensed to form the bond. So we have different types of oligosaccharide. The first one is disaccharide. The name only tells you di, bi, these things means two. So when there are two monosaccharides that came together and formed the oligosaccharides, we call it as disaccharide, such as sucrose, which is made by the condensation of glucose and fructose. Maltose is made by the condensation of glucose and another molecule of glucose. Then we have lactose, which is made by the condensation of glucose and galactose. Lastly, we have trehalose, which is made up by glucose alpha 1 and glucose alpha 1. Another like two molecules of glucose again joined together, but the alpha carbon, the first carbon. Clear, students? These are the various examples of disaccharides. Furthermore,
Difference between glucose and glucose alpha 1.
see, this glucose alpha 1 refers to that the first carbon of the glucose is forming the bond in both the cases. Understood? The first alpha carbon. Okay. Whereas the normal glucose, C14 linkages. Okay.
Okay, ma'am. Got it.
Talking about trisaccharide, tri means 3. So now these sugars are made up of 3 monosaccharide units. So such as example is raffinose, which is made by the condensation of glucose with fructose with galactose. 3 sugars coming together making the trisaccharide such as raffinose. And then we have tetrasaccharide, which are made up of 4 monosaccharide units. So stachyose. Examples, it is made up of 2 molecules of galactose, 1 molecule of glucose, and 1 molecule of fructose. Understood, students? Any doubts? So this was about the oligosaccharides. Now, mostly all the monosaccharides, they are reducing sugars. Their nature is reducing in nature. What is reducing nature? Meaning they can reduce the copper di-positive ion to copper monopositive ion from Cu2 plus to Cu plus ion. Okay, this is their reducing property. For example, there is sugar molecule, you've taken sugar molecule and you've taken copper ion. So that sugar molecule will react with the copper ion and reduce it. Okay, it will make it lose its charge. So that will become Cu plus. Okay, this property of these sugars, like their reducing nature, is used in checking of glucose in the urine or in the blood sample. So why are they able to do this reduction process? It's because they have either a free aldehyde group. Free means not bonding with anything. For example, like this, and then there's CH, which is not bonding with anything. So we say that it is free. Same way, the ketone could also be present and we'll call it as free. Most of the monosaccharide, you can say all of the monosaccharides, they are reducing sugars except for one, that is sucrose. Remember this, again, very important. Sucrose is a non-reducing sugar. Why? Because it doesn't have any free aldehyde or free ketone group. Did you people get it? Any doubts?
Ma'am, can you explain one more time?
See, I'm saying that the monosaccharides, they are called as reducing sugars. The reason why they are called as reducing sugars is because they can reduce the copper di-positive ion to copper monopositive ion, meaning from Cu2 plus to Cu plus ion. Why can they do that? They can do so because they have a free functional group. Either they will have a free aldehyde group or they were going to have the free ketone group present in it. Understood? So most of the monosaccharides, they are reducing sugars. But there's an exception. We have sucrose, which is a non-reducing sugar. The reason why it is called as non-reducing, because it does not have any free aldehyde or ketone. That is why it cannot reduce copper to copper, like copper dipositive to copper monopositive. Understood?
Okay.
Now we have polysaccharides. Poly means many, right? Now these are those polymers that are made up of several monosaccharides. Not one, two, it's like sometimes many. So these are large molecules. So we put them under macromolecule condition or macromolecule category. So now here also the linkage is glycosidic linkage between all the monosaccharides. Now, mostly in a monosaccharide, the right end, mostly in a polysaccharide, the right end is the reducing end because it either has a free aldehyde or free ketone. But the left hand side is the non-reducing end. It has both the kinds of end present. Now, depending upon whether, like see, there can be two examples. For example, this is polymer number 1 or polysaccharide 1. This is polysaccharide 2. You see the difference in between them?
Yes, ma'am.
What is their difference? The units that they are made up of, right? In the first situation, you see only circles, whereas in the second situation, you see all sorts of shapes. So depending upon whether the polysaccharide is made up of only one type of monomeric unit or different types of monomeric unit. So the polysaccharides, they are of two types, homopolysaccharides and heteropolysaccharides. Now, homopolysaccharides, they have only one type of monosaccharide unit or monomer. Examples for which are glycogen, starch, inulin. Remember, it's not insulin. It's a different thing. Inulin, then there's chitin and cellulose. All of these are the various examples of homopolysaccharide. Homo means same. So those polysaccharides that are made up of a single type of monomeric unit or a single type of monosaccharide, we have homopolysaccharide. For example, cellulose. Where is cellulose found, students? Give me the right answer. There. Give me the right answer. Where is cellulose found? This is the most basic answer I could have, like question I could have asked from you. And this should come very instantly. Students, where is cellulose found? Everyone knows this. You people have studied cell chapter also. No one.
I'm not sure, but is it cell wall of plants?
Is it?
Cell wall of plants?
So you should be sure, na? Everyone should be sure for this. Cellulose is what the cell wall of plant is made up of. It's the composition of a cell wall in plant. What is the bacterial cell wall made up of? Very bad. What is it made up of? Huh?
Polysaccharides?
Peptidoglycan.
Oh.
What is the fungal cell wall made up of?
Chitin.
Chitin. These things are very basic things. People should remember this. So, homopolysaccharide, we have cellulose, which is a homopolymer of glucose, and it is found in the cell wall of plants. Then starch and glycogens are also homopolymers of glucose. Starch is the storage food material of plants, right? And glycogen is the storehouse of energy or the storage of animals. Then talking about what is inulin. So it is a polymer of fructose. Okay. And it is a storage polysaccharide. Where is it found? Mostly it is found in the roots and the tubers of dahlia plant. Now this inulin is a plant product. Okay. So just like if you take this or we eat it, we do not have the enzyme to digest inulin. So many a times this inulin is used to test the kidney functioning of our body because it cannot be metabolized in the body. So the kidney will in the urine, inulin will be found untouched or unbroken. So this, if the kidney is functioning well, then the inulin will be found in the urine. If it is not functioning well, then the kidney will not produce inulin. This is used for kidney function test, KFC. Clear? Then we have chitin, which is the homopolysaccharide of N-acetyl glucosamine. And where is it found? It is found in the cell wall of fungi. This is the ordinary answer. You know, if I asked you where is chitin found and you people said cell wall of fungi, this is the ordinary answer. Where would have, I might have gotten up from my seat is when you people might have told me, ma'am, that it is found in the exoskeleton of arthropods. Then I would have been like, I'm done. But no one is studying. I can see that. The homopolysaccharides are clear to everyone.
Yes.
Yeah, ma'am.
Then we have heteropolysaccharides. Now, heteropolysaccharides are the ones that are made up of more than one type of monosaccharide units or more than one type of monomers. So we have peptidoglycan, we have hyaluronic acid. These two examples are there. You don't have to learn their monomeric units. You just have to know what is the example. So any doubts up until now, students? So we have completed carbohydrate part. Any doubts in that?
No, ma'am.
No, ma'am.
All right, now you people note it down, then we'll move on to amino acids. And try to write a little fast.
Done, mom.
Everyone's done.
Yes, ma'am.
All of you are done, students. Shall I scroll down?
Yeah.
Done, students. Shall I scroll down?
Yeah, mom. Yes,
No,
ma'am.
not done.
Now we can scroll more because we already finished writing the first part. No, ma'am, you can scroll it down.
Excuse me, ma'am.
Yes.
Ma'am, can you explain what are metabolites?
Metabolites.
Yeah.
See, basically, when we talk about metabolites, these are the molecules that are produced as sort of like, imagine it like an intermediate of any kind of metabolism process, meaning the result of metabolism is metabolite. Clear? So those metabolites that are needed to stay alive, that are directly involved in very important functions of the life like growth, development, reproduction, all of that. So those metabolites are primary in nature. Okay, they're very important. Other than that, the ones that are not directly involved, but they have some sort of function like defense, some sort of function like signaling. So we call it as secondary metabolites. Understood?
Yeah, ma'am. Thank you.
I'll write it here.
Done, ma'am.
All of you are done?
Yes, ma'am.
Yeah.
Done, ma'am.
Done.
Done, ma'am.
Everyone's done?
Yeah, ma'am.
All right. Any doubts up until now, students?
No, ma'am. Done.
Done, mom. Done, ma'am.
10 students.
Yes.